Okay, Maya, are we good to go?
Yep, we're good.
Okay. Well, everyone, thanks for joining. Welcome to our second Hot Papers webinar. And my name's Ian Lanza, and I'm the chair of the ADA Exercise Physiology Interest Group. And on behalf of my colleagues in the interest group, I'm delighted to be the one that gets to welcome all of you, those of you who are joining live, as well as any of you who are accessing this archived content on demand. This is a relatively new activity that we've done as an interest group. The idea here is to really showcase some exciting new research that we think will be of broad interest to the members of the interest group. And the format is designed to really be kind of symbiotic in that authors have a platform that they can promote their new research. And then the attendees get a knowledgeable concierge who can really walk us through the main findings of the papers. And this would be the author of the paper and an opportunity to engage in discussion. So the format's supposed to be brief and focused and informative. We hope to have a few minutes for questions and discussion at the end. So if you're joining live and you want to save your questions, certainly can save them for the end, or just type them into the chat and I can make sure I ask them at the end. So today's hot paper is titled Muscle Mitochondrial Remodeling by Intermittent Glucocorticoid Drugs Requires an Intact Circadian Clock and Muscle PGC1 Alpha. And this was published last month in Science Advances. And so today the presenter is Matteo Quattrocelli, who's the corresponding author of the paper and currently an assistant professor at Cincinnati Children's Hospital Medical Center. So thanks for joining us. Really appreciate you taking the time to take part in this webinar, and I'm going to mute myself and turn it over to you.
All right. Good afternoon, everyone, and thank you, Ian, and everyone on the ADA exercise group for giving me the opportunity of presenting this work. So I'm Mattia. I join you today from Cincinnati. And without further ado, I will start presenting the findings if the computer complies. All right, so we have been interested quite a bit into the timing of the effect of glucocorticoid drugs on muscle metabolism, mainly on two dimensions, on two time dimensions. On one hand, the frequency of intake. This is past work, for instance, that we reported in the previous years in the dystrophic muscle, where we found that contrary to the pro-wasting effects of once daily intake of these drugs, an intermittent frequency of this intake of these drugs, for instance, a once weekly intermittence, actually promotes muscle metabolism. In this paper, we are actually assessing another dimension of timing of this drug intake, and that is the circadian time of intake. Is there a difference in the effects of these drugs on muscle metabolism when we dose them at different times of day? And this question starts from a rather scattered rationale in literature that suggests the possibility that the glucocorticoid receptor, which is the main receptor of these drugs, acts differently according to the ligand-mediated solicitation in different times of day. The glucocorticoid signaling axis is strongly linked to circadian rhythm, regulates the circadian rhythm, and in turn is regulated by the circadian rhythm. And this is, for instance, due to the endogenous glucocorticoid rhythms. In mice, for instance, that would be corticosterone. In humans, that would be cortisol, which, for instance, encumber the glucocorticoid receptor in one period. In this case, in mice, that would be during the dark phase. And then in the late light phase, again in rodents, the glucocorticoid receptor is somehow inhibited or counteracted by other circadian factors, for instance, clock. However, there is some scattered evidence in literature suggesting the fact that in the early daytime, in the early light phase in mice, the glucocorticoid receptor is particularly susceptible to exogenous ligand activation. So that would be to activation from drugs, for instance, prednisone in this case. And so we asked the question of whether a different time of intake of this drug is linking in a different way, possibly in a different quantitative way, the GR activation to muscle mitochondrial health. And we focus on muscle metabolism here because muscle is still one of the main tissues in our body dedicated to energy production and, frankly, energy expenditure. And so in a first experiment, we treated wild-type mice with intermittent prednisone, because as we found in the past, intermittent prednisone was this pro-metabolic treatment, restricting the dosing either at the beginning of the light phase, that is CT0, so lights on at the beginning of when the lights go on, or at CT14 in this case, so that is when the lights go off in this experiment, in the facility with these mice. And so compared to vehicle, light phase treatment increased muscle performance, shown here, for instance, as workload on a treadmill, improved muscle endurance, shown here as fatigue in an in situ tibialis anterior muscle analysis. This correlated with an increase in overall body-wide auxiliary metabolism, shown here is VO2 ANCOVA against lean mass. And this correlated with a gain in muscle NAD, as well as muscle basal oxygen consumption rate of muscle tissue. And what was really interesting in these experiments was that when we repeated these with dark phase treatments, these metabolic effects, or frankly, even these muscle function effects, were completely lost. So this led us to ask, is there a difference in these parameters due to a change in overall circadian rhythm in mice, in overall circadian behavior in mice? And the answer seemed to be no, because we could not find differences in rest phase or active phase corticosterone. Again, this is the endogenous main glucocorticoid in mice. The rest phase is in mice is the throw of corticosterone, and the active phase is the peak of corticosterone in mice. And in metabolic cage analysis, while we found an increase in the VO2, especially in the active phase, we could not find differences, treatment differences, or even circadian time of treatment differences with regards to spontaneous locomotion or food intake. We then result to ask whether the circadian rhythm, and specifically the circadian clock, and specifically the molecular clock, is somehow involved in these effects. You know very well that the circadian clock ticks the circadian rhythm in every cell, in every tissue. It is based on a rather complicated molecular machinery. And for this talk, for this work, I want to draw your attention on BMAL1, which is one of the main factors involved in the activating clock forearm, which has a peak activity as a factor itself, as an epigenetic factor itself, in the light phase in mice. And so we, considering that we were finding these treatment effects restricted to the light phase treatment, to the light phase injections, we asked whether BMAL1 was involved in these effects. And so we gathered litter mates from BMAL1 wild type and BMAL1 knockout mice, transgenic mice, and we treated them again with the light phase restricted treatments. And as you can see, while in the BMAL wild type mice, we were able to replicate the treatment effects on muscle NAD, muscle oxygen consumption rate in the presence of either glucose or palmitate. Again, the effects on muscle performance, fatigue, and body-wide VO2. These effects, once again, were significantly blunted or lost in the genetic absence of BMAL1. And because both the glucocorticoid receptor and BMAL1 are epigenetic factors, we decided to profile what goes on with these epigenetic factors in muscle with a light phase specific, with a ZT0 specific pulse of prednisone. We performed CHP-seq in muscle, at 4 hours after the CT0 drug injection. We profiled, for instance, GR epigenomic occupancy in BMAL1 wild type and BMAL1 knockout muscle. And the ChP-seq was validated by unbiased motif analysis. You see that the top ranked motif was the glucocorticoid receptor element, the GRE. A principal component analysis of the peak profiles, clustered the samples according to treatment as well as to genotype. But when we analyzed the genome-wide GR signal on the canonical GRE motif, we did find the first surprise that in the BMAL1 wild-type muscle, the drug was expectedly increasing binding of the GR on top of the canonical GRE motif. Whereas in the BMAL1 knockout muscle, this binding, this increase in signal was significantly blunted. It was still present, it was still there, but the effect magnitude you see was significantly lower. And this is just a heat map representation of these quantitative curves. And then we profiled the BMAL1 genome-wide occupancy in muscle, again at 4 hours after light phase injection. Again, the data sets were validated with motif analysis. The E-box, which is the canonical binding site for BMAL1, was the top motif scored here. The PCA, the principal component analysis of the peak profiles, clustered again the samples according to injection, to treatment, vehicle versus prednisone injection. And here we found another surprise, that BMAL1 is not a drug binding is not a drug binding factor, does not bind directly prednisone. However, the activity of BMAL1, the occupancy of BMAL1 on the canonical E-box motif in muscle was indeed increased by a light phase injection. And even more intriguingly, overlaying the data sets, we found that the GR signal on the canonical BMAL1 binding motif was increased by the injection, by the light phase treatment, as well as vice versa. The BMAL1 signal on top of the GRE motif was increased. This led us to hypothesize that there's basically the light phase drug injection was stimulating a transient interaction between the two factors in muscle. And this was further supported by the fact that, again, overlaying the data sets, we found an increase in GR BMAL1 peak interdistance between 0 and 300 base pairs and 300 and 900 base pairs, which are generally regarded as peak interdistance of factors either overlapping on top of each other or closely interacting with each other. We then performed a ChP-seq for RNA-POL2 as a marker of transcriptional upregulation or downregulation. And we filtered all the genes that were having a difference, a difference in transcription, a difference in RNA-POL2 binding to the transcriptional start site, dependent on BMAL1. And among those genes, we categorized the genes based on the enrichment in their promoter for both the GR and the BMAL1, again, at 4 hours after a light phase injection. We filtered these genes for geotherms of muscle metabolism and circadian metabolism. And the top two genes that came up were NAMPT and PGC1 And indeed, you see that both NAMPT and PGC1 showed an increased GR binding and BMAL1 binding in their proximal promoter. And this increased binding of the GR was lost or blunted in the genetic absence of BMAL1. And why are these genes important? Because NAMPT encodes NAMPT, the enzyme that produces NAD, or one of the enzymes that produces NAD, and definitely the rate limiting enzyme. And PGC1 is the known agonist of mitochondrial biogenesis and mitochondrial function. We then asked whether in muscle these epigenetic changes were actually resulting in transient changes in protein abundance, and they were. So these are Western blots from muscle lysates, assayed at 0, 1, 2, 3, 4, and 5 days after a light phase specific prednisone shot. And indeed, you see that there's a transient upregulation of both NAMPT and PGC1 alpha at a total level. And intriguingly, also the PGC1 alpha shows a transient decrease in acetylation level, as well as a transient increase, a coordinated transient increase in phosphorylation level. This is kind of important because indeed NAD and NAD, gain of NAD availability and PGC1 alpha are closely related. PGC1 alpha is deacetylated by the NAD dependent sirtuins, and if it is deacetylated, it's primed for activation by phosphorylation. And indeed, we asked whether PGC1 alpha was driving the mitochondrial remodeling downstream of light phase intermittent prednisone, in this case, light phase pulsatile prednisone, a pulse of prednisone. We generated inducible muscle restricted PGC1 alpha knockout mice, where we could remove PGC1 alpha only in the adult muscle and only after tamoxifen treatment, in order to avoid developmental or early life adaptation to the lack of PGC1 alpha. After PGC1 alpha removal in muscle and washout, we again repeated the experiment with a light phase prednisone pulse. And then we tested again the mitochondrial abundance through mitochondrial DNA over nuclear DNA ratio, not shown here, as well as through mitotracker fluorometry in tissue. And indeed, we saw that while in the PGC1 alpha wild type, the light phase prednisone was increasing mitochondrial abundance, this effect was lost in the absence of PGC1 alpha. Analogous trends were obtained with regards to basal muscle tissue oxygen consumption rate, again, both in the presence of either glucose or palmitate. And these trends were also appear to be mitochondrial specific, or at least dependent on the actual rate of respiration of mitochondria, because we isolated mitochondria from muscle and we measured their respiratory control ratio, which is a quantitative measure of how much the mitochondria are respiring. It is indeed the ratio between state 3 and state 4. And while the light phase prednisone was increasing the RCR, the respiratory control ratio of isolated mitochondria in the PGC1 alpha wild type muscle, this effect was lost when the muscle PGC1 alpha was removed, was ablated. Following up on the idea of a transient interaction between the GR and BMAL with a light phase specific, with a ZT0 specific prednisone pulse, we performed other analyses on the nuclear translocation, as well as the protein-protein interaction of GR and BMAL1 in myofibers, in isolated myofibers, as well as in muscle tissue. Here I'm showing you images of 3D confocal live imaging on isolated myofibers after a ZT0 prednisone pulse. You see that the GR migrates to the nucleus, as well as the BMAL1, and this is quantitated here. And in the paper, you will find a host of other assays showing that indeed there's a transient protein-protein interaction in muscle. There's an enrichment of these factors in the nuclear fraction with this light phase specific injection. And intriguingly, these effects were negated by a reverb alpha overexpression, further underscoring the importance of the circadian clock in this. Reverb alpha is another molecular factor involved in the circadian clock, and reverb alpha generally counteracts the BMAL1 translocation to the nucleus. It's part indeed of the repressive arm of the clock complex. To summarize this with some takeaway ideas, some open questions, and some interesting clinical developments of this. So the take-home message of the paper is that the time of day of glucocorticoid treatment, of glucocorticoid drug injection, does remodel muscle metabolism in a different way according to circadian time of intake. Specifically, in mice, a light phase, an early light phase injection of prednisone positively remodeled mitochondrial metabolism through GR and BMAL1. Open questions that we are addressing and that will be important to address is what is the role of the immune system and what is the role of circadian glucocorticoid, circadian time of intake of glucocorticoid drug on immune system regulation. What is the interaction with the other clock components, and what is the role of this molecular circuitry in glucose uptake and use by muscle. A last bit of early exploratory clinical development that I'm presenting has just been published in J&D just a few weeks ago, is that these are initial results from an exploratory open label trial performed at Northwestern University on non-Duchenne dystrophic patients with once weekly intermittent prednisone. And the patients, partially based on the ongoing studies that I presented to you today, these patients were advised to take the drug at any time between 7 and 9 p.m. in a first exploratory effort to translate the early light phase treatments in mice to humans. And after 6 months, these the DEXA scans showed a gain of lean mass over the baseline, but no changes in fat mass. And there was also an initial increase in the 6-minute walk test, as well as in the 10-meter run test, which are two ways of assessing muscle function in the clinics. So again, this is an exploratory open label clinical trial, so it's a very early assessment and it's very rough translation. That said, though, I think that these data somehow show that there is some at least some possible clinical aspect to this, and we will see this more better in the future. With that, I thank you for your attention. I thank the organizers for the opportunity. This is our lab. I thank all of our collaborators and our funding agency. Again, all of you for your attention, and I really would like to take questions and respond to them. Thank you.
Thanks, Mattia. That's great. We have time for a couple questions, and I'm not sure how we're set up here on Zoom, if people can unmute and ask, or if they have to type it in the chat. But I'll start off with a question or two that I had. You know, what's fascinating is, you know, when you look at the whole body VO2 data, you know, you're seeing that increase with the prednisone pulse, right? Is that VO2 peak, or is that sort of resting VO2?
The effect was present both in the rest phase as well as in the active phase, but the higher effect magnitude was in the active phase. Okay.
I was trying to tie that back with energy expenditure, just to get a sense of, is sort of whole body energy expenditure higher in these mice?
Yes, we found these are evidence, these are data that we're publishing in another paper that is coming out in the next days. That indeed in wild-type mice, and especially in wild-type mice fed a high-fat diet, so in conditions of diet-induced obesity, we do see an increased energy expenditure.
Mm-hmm. Got it. So I was thinking, is you know, can you trace that back to a mitochondrial phenotype of maybe energy, so inefficient mitochondria contributing to increased energy expenditure, but then your RCR values are actually higher, which would suggest that maybe there's less proton leak and maybe more efficient coupling of mitochondria. So that's interesting. Yeah, it's good to know you have another paper coming out looking at energy expenditure. That'll be interesting to see. I see Jane wrote a question in. Says, what do you think are the clinical implications of the normal cortisol rhythms in the morning?
Yeah, that's a good question. Indeed, I want to stress the point that all these findings and also the analysis and the statistics that we ran in the paper were mainly focused on the drug effect. so on the exogenous glucocorticoid delivery. The implications with regards to the endogenous steroid rhythms have to be investigated and have to be better addressed. That said, though, in the paper, we did not find with intermittent steroids, this is very important, with intermittent glucocorticoid regimens, so once weekly prednisone regimens, we could not find long-term distortions of the, or quantifiable distortions of the endogenous circadian rhythm in mice. With regards to standard parameters of circadian rhythm and endogenous corticosteroid levels. In the exploratory clinical trial results that have just been published, I could not see in the publication, I did not see reports of, let's say, or of even anecdotal reports by the patients of sleep disruptions and so on. Again, these are at present, these are just suggestive indications, but better a better investigation of this needs to be addressed. I think there was another question in the Q&A.
I'm only seeing James.
So I've opened a Q&A box and there's a question from Inna. When is the best time to exercise then? Okay, that's a good question. I want to stress that the point that this study was performed in the absence of an exercise stimulation. We did not exercise the mice. We did not put a wheel in the cage, or we did not exercise them, on the treadmill for weeks and months as an exercise intervention. And this was done on purpose in order to see whether the epigenetic effects of the time-specific drug intake were regulating muscle metabolism per se in the absence of exercise. For the role of exercise, and especially for the role of circadian rhythm in exercise, I would reference to quite some literature that is now present in the field from other and better groups at investigating this. There are now several reports addressing the differences in the exercise effects on overall metabolic function as well as muscle function with regards to different time of day. That said, though, it is a good question because indeed how the exercise and exercise activity and so on, especially as an intervention, copes or conflicts with the circadian specific trends that we're seeing with glucocorticoid effects, this is an open question and we are looking into this. And then I see another question from Jane. We tend to give prednisone in the AM for clinical conditions, so we do not suppress normal profiles. Should we rethink this? Jane, this is a good question. I cannot answer this with experiment-based evidence because we are indeed addressing some of this, and this is some. To be clear, the interaction between the HPA axis and circadian time of glucocorticoid intake will be a rather difficult topic to pick apart. That said, though, what I can tell you is that, especially from the experience that we have with mice, it looks to me like there's a combination of the circadian time of intake as well as dosing frequency. Once daily glucocorticoid intake will probably suppress the HPA axis and will result in adrenal suppression and metabolic stress and so on, probably even, especially in the long term, or probably even regardless of the circadian time of intake. Whereas with the intermittent intake, there is probably an opportunity to sort of leverage the circadian time of intake and therefore maximize the benefits over the side effects. The problem is that all of this is a speculation at present. and further work needs to be done in this, especially in clinical settings. The open question is that, and also an important ancillary open question is whether intermittent intake as well as, circadian specific or circadian gated intermittent intake of glucocorticoids needs to be tested with regards to efficacy for the primary prescription of the drugs themselves. For instance, is a circadian specific intermittent glucocorticoid intake still efficacious with regards to the primary treatment of a number of immune conditions, or arthritis, or RA, and so on? And these are questions that are still open.
That's great. Looks like we're out of time. I encourage people to reach out to you with additional questions, especially people who are streaming this on demand, and I probably can get your contact from the paper as corresponding author. So thanks for joining us. Really appreciate it, and we'll see you next time.
Thank you again for your time and the opportunity.